Okay, so first of all, thanks for the invitation. It's great to be here and to be with this community. So, since the theme was about extreme statistics, I went into the previous work that we did some years ago about how we encounter extreme statistics. You know that we were interested in applying extreme statistics, and this was in a problem in cellular biology that I will explain, and how these extreme statistics are actually constrained by the players, which mean the molecular organization inside the structure called a dendritic spine. So extreme statistics, I don't have to explain because, after being one of the last speaker, the last day, you have all understood that the statistics of the first one has nothing to do with the statistics of the population. And if you do the rescaling, you find out that the mean, or the first one to arrive, falls into these three types of distribution. So at this point, we can say, look, if there is some universal distribution, it means that we have encountered something that is universal, and so the underlying biology might not be that interesting. So in that case, you say, okay, you capture the physics, fine, you can fit your curve, but how much does that tell you really about how things are working? And this is this aspect that I would like to share with you. So statistics of the extreme, we will be interested in the first one to arrive, I will explain, and how can this be used to do a reverse engineering problem in molecular and cell biology. If I have time, I'm not sure, I want also to explain another type of work that we started about 10 years ago, is how extreme statistics define the time of senescence. We're all interested in time, but it's interesting to see that the time can be connected to the first telomeres that reach a certain threshold, which define the moment where the cells stop to divide, which comes to an extreme statistic question. So if I have time, I will discuss this. So first part of the talk, I'll explain this reverse engineering. Then I would like to go to a bit of theory, how you calculate the time for the first one in different conditions, when you can switch, when there is killing, and especially what happens if instead of putting all molecules in one place, you have a distribution, which in that case, instead of 1/log, you can change, you can have power laws because of the tail. So if I have time, I'll discuss this. And then senescence, time to stop division. I'll try to do my best, but if I reach the first topic, I think I'll be very happy. Okay, extreme statistics are making rare events probable. So, and also a fundamental question that we have been interested in is what defines large N? What is N large? N going to infinity? It doesn't work like this in biology. N is fixed. We had this example a couple of days ago about neurotransmitter. Why you have 3,000 neurotransmitter, 1 million in a single vesicle when you have neurotransmissions? So we thought a lot about this kind of question. This has to be related to the geometry. Because you have to find something in a synaptic cleft will have such a dimension, and this constraint in a very complex, maybe evolutionary manner. So here I would like to just give an example of fertility. As you know, the first sperm that will arrive is going to be the one that will be able to fertilize. So this is some data from that we collected many years ago, where you see the sperm are not random. They have this persistent trajectory. We did the model, look at if you now reconstruct a structure and put tens of hundreds of thousands of sperm having this model that can be reflected with a random angle here on the surface, and how long it takes to find here one of these ends. And you see that the optimal trajectory is the white one. The optimal trajectory has nothing to do with the trajectory associated with one of them. And this is associated to, you have to minimize here some action, and this is related to the large deviation principle, although here it comes, we can derive it from somewhere else. Okay, what is interesting is that if you divide by 4 the number of sperm, although there are hundreds of millions, you get infertility. It doesn't work. So numbers, precise number matters. Yeah, and if you're interested, we publish all of this and this kind of question. I was thinking, about 10 years ago. Yeah, and so finally, to explain a bit of the extreme statistic, you can ask, what's the connection between the number of sperm and geometry, which is the size of the uterus? Is there a relation? Because this should be two things that have no connection. The sperm doesn't know where it has to go. That's why you make redundancy. That's why you make many of them, many of the same object, in order for one of them to find. And if you look at, you try to make some, this is completely heuristic. You take the uterus, take the number of sperms, and if you want to fit something, you get this kind of law, which is purely empirical, and you try to make sense of this in terms of the numbers versus the geometry. What does this teach us about fertility? Today, I have no answer to this question. What is the principle of adaptation between the number and geometry? And so we discuss a bit this with actually no answers at the moment. Okay, let's start the talk. I would like to discuss today extreme events at a molecular level. So this is the brain. The brain is made of one of most relevant components, which are called neuronal cells. Neurons are connected through a structure called a synapse. And we are interested, the synapse is made of two boutons, of one button with two elements, which is called the presynaptic terminal, postsynaptic terminal. And I'm interested in one of them, which is called the postsynaptic terminal. And we would like to understand how the architecture inside defines signaling. Okay, so this is a neuron. So you can see this structure. This is a structure we'll be interested in. This is called dendritic spines. And they play a multiple role, especially in signaling. So this is pre-post. When an action potential comes, you have vesicles that are going to be released. These neurotransmitters are moving by diffusion. They interact with one of the receptors. And when you have enough of them, a signal comes, such as calcium. And then the question is, what is calcium doing? What defines the time course of calcium in these domains? Okay, so calcium comes in. And now you can ask the question where calcium is going. Is calcium interacting? Is calcium finally flowing directly? And you have the same amount of calcium here that escape there. Okay, I have to give you a little more background. So this is now, we have to enter in opening the box. Inside the structure, there are organelles. And one important organelle is called the endoplasmic reticulum in blue. This is a complex structure that can sequester calcium and does many other things. So here, this is an example of electron microscopy where you see this part of the ER, which is called a spine apparatus, which is very well isolated. It doesn't connect necessarily to the rest of the ER. So now you can experimentally have an indicator that will tell you if yes or no, you have one of the structure in a spine. Sometimes you don't have. And so these are examples where you can have this structure inside the dendritic spine. Okay, so now let's move on into some of the signaling. So what is commonly seen in textbook is that I will come to the specific description. You have here receptors that are able to release calcium from the internal spine apparatus. This is called ryanodine receptor. There's another one called IP3 receptors. And you can have calcium that enters here that can be moved in by Cerca. So there are two possible directions. It can go in or it can go out, according to these two types of receptors. Okay, now this is the key of the talk. If you're on the telephone, it's the moment that it just... Because we spent 2 years, 3 years just on understanding these single experiments. Old-fashioned way to do science, just look at the experiments, what does it tell us? So I'm going slowly. Now you have a machine. So this is the, whatever the spot color is, this is purple. So you induce calcium in the head. And you ask, what do you see? What do you see? You induce a mechanism that will induce calcium in the head, and you ask, what do you see in the head, and what do you see at the base? Answer. In the head, you see this peak of calcium, and at the base, you see this curve, which Why it is puzzling? Because after 5 milliseconds, you see a significant amount of calcium at the base. Now this is, you see the green, it means that there is a spine apparatus. Now if you do the same experiments, but you engage calcium here at the base, well, you see increase of calcium at the base, but you see very little in the head. So this already said that there's some kind of asymmetry. Head, base. Head, this is the head, the round, and the base is here. The head, yeah, sorry, the head of the spine. So this is this head here, these compartments, and the base is here. Just above. Now, you repeat the same experiments in the head and in the base. No spine apparatus. What do you see? You see that you have, again, this calcium increase here. Nothing at the base. Now, if you repeat it in the opposite direction, you see the same as here. Something at the base, but nothing in the head. Question, how can we understand this? Now I'm going to accelerate time, and instead of going through these 3 years, in 10 minutes, I will tell you the answer, how it works, and how we found it out. Yeah, because this is fluorescence. Doesn't matter. Okay, so this... No. Only in some. This is a very good question. Only in some of them. They
behave functionally
different. This is already.
No, this is dynamic. Size, all those things having to do with.
Yeah. So there's a huge literature. So when you have a spine apparatus, the spine is much stable. When they are not, they are much more dynamics. There's a mushroom between steady spine. It's a fundamental difference. And that's right.
Okay.
That's right.
That's right. I'm going to believe you.
Yeah. No, this is a huge literature. People still are interested in understanding, why you have this spine apparatus here, because we'll see it has a fundamental function. I'm not going to discuss this, but it's also very important for metabolism, because then it is associated to mitochondria. But that's another talk. Maybe next time I'll discuss this. Okay, so now let's dive into this. Now you do a simulation, Brownian simulation. You have this. You recapitulate the geometry, and when you look at the calcium here and the calcium at the base, note that this is of different scales. This is blue is hundreds, and here is the number of calcium arriving here at the base. So it's just few ions, which is incompatible with what we see here. So simulation, little increase. We have fast, it propagates quickly in the order of few milliseconds, but it's nothing. So this cannot recapitulate this. We are more in this stage. Only one place. They can only here enter and exit here. That's this simple simulation. Yeah, this is the source. This is the calcium engaging. This is the red cross. It starts here, and you ask where it goes and how long it takes. So to go into this into more details, if you look at this is all the type of experiment, but this is very precise. You can look in the fluorescence what is the time course of something diffusing. And you see this is of order of hundreds of milliseconds, which has nothing to do with a few milliseconds that I just described to you. Then you can go into analysis. So why you develop asymptotic formula is to see how the geometry is actually defining the time it takes to go from here to here. It has to do with the radius here, the size here of the neck, the length of the neck, the diffusion coefficient. So this allows to connect time with geometry. And again, when you take this, plug the formula, do the simulation, you get hundreds of milliseconds, which has nothing to do with the few milliseconds that we just described. Because, calcium has been understood experimentally and theoretically as diffusing. This is what you can see in the literature. So this is very well known that calcium is diffusing. No, this is not the scale we're interested here. Here we're interested in micrometer. We are inside the cell, calcium goes in, and when you look at fluorescence and the calcium typically diffuses, this is a very good approximation. Okay, so now let's involve now the spine apparatus. So, which means that somebody asked the question, maybe calcium doesn't simply go through. Okay, let's investigate this. So you have calcium bound to buffer, but buffers takes much longer time. It's tens of milliseconds. So we can neglect buffers here for the purpose here of the time scale. But then calcium can go inside through the theca and can be released by ryanodine. At the time scale we are interested in, there's another receptor. We can neglect IP3 receptors because it takes much longer time scale. So then again, we are interested in adding the spine apparatus, like a spine in a spine, where you have now compartments, and now you have to position the player, which are the ryanodine that allows calcium to go out, and cerca that allows calcium to go in. Okay, so now this can affect calcium, so now you need a model. Again, I'm going very quickly about the description. Cerca pumps, the way to describe it is when a calcium goes at a molecular level, you need a second calcium, and then they are translocated. For ryanodine receptor, when you have two calcium, then a certain number of them can be released. And the time of translocation is about hundreds of milliseconds. Ryanodine open quickly and release a certain number of calcium, which you can fix from few tenths. And there could be a possible delay, but of the order of milliseconds. So now what is interesting is that ryanodine, they come in cluster. This has been documented in the cardiac myocytes. So they come in cluster, which means that many of them are together creating a nanodomains. And then you can model this as these nanodomains. And why this is interesting, because when calcium here will trigger the first one, you can have a release of calcium that will trigger the next one. And this is called calcium-induced calcium release. It's basically an amplification. So after a first release, ryanodine cluster amplify this calcium signaling quickly. Okay, so now we started to do, again, the framework of very naive Brownian simulations where you allow your calcium to move with a diffusion coefficient, and you start with thousands of ions in the head to start with. This represents about 0.4 micromolar. And now the question is, where are we going to position the two types of molecule, the receptors and the ryanodine receptors and the cerca pump? Because we looked in the literature, there was no evidence where they are. They say it's everywhere. Okay, so this we started to put some cerca here at the top and ryanodine at the base. Why? Just to try. So this is what we started with, and let's see what the simulation says. Release thousands of calcium here. Now suppose you have no ER. So this is what happened. This is the green curve, nothing, no much ions arrive here. Then we have the cerca, but no ryanodine receptor. Again, nothing happened. Then we have no cerca, no ryanodine receptor, nothing happened. And suddenly we put ryanodine, and now they can release. You can play with how many ions they release, 3, 4, 5, and after 5, doesn't matter, because now you start to see something here. What does it mean? It means that when you have these ryanodine receptors, you have this amplification that leads to this very quick response, although diffusion said it's not compatible. So you have calcium-induced calcium release, fast calcium increase at the dendrite in the compatible timescale. Then what we did, we started to do into the math, looking now because what exactly you need? You need two ions, not one, two ions to arrive to a single ryanodine receptors. And when we look at the distribution of time for the first one to arrive, this is how we connected to extreme statistics, we indeed found that they were compatible with the timescale of the experiment, suggesting that the first ion to arrive are compatible with the fact that you see increase of calcium. So where calcium comes from? Calcium doesn't necessarily from this, doesn't come from the flow coming from the head to the base. It means that the first one comes, it opens calcium, induced calcium ring through the ryanodine. You have an amplification because they are in a group. And most of the calcium that you see at the base is due to the release triggered by the first one to arrive. So, indeed, when we position ryanodine at different places, at the base, at the head, we splitted them in different conditions, what we saw is whatever the splitting here, there is only one that were working very well, is when they are at the base, confirming the idea that you need this clustering to create this amplification. Yeah, and this is when you start with 50, 100, you increase the number of initial calcium, and you see only this peak, which is relevant. So putting ryanodine everywhere doesn't work, it's not effective, so they should be only at the base. And now, 5 minutes, okay. I knew it would only be the first part. It doesn't matter. What is important is to connect. Okay, then we went to our experimentalists and look, nothing in the literature. Is it true what we said? So they did these fantastic immuno experiments. They say, all right, let's see for the spine that have a spine apparatus, where are Serka pump and where are ryanodine receptor? And ryanodine receptor, as you see here, were mostly below the spine, while the Serka pump was mostly in the head. So they confirmed this using super resolution microscopy, where indeed we had the same effect. Suggesting that here we use in some sense the extreme statistics events to explore the different organization and found out that there was only one possible organization that will underlie this mechanism of signaling by the extreme. Okay, so then we revisit the textbook that ryanodine should not be in the head. They should appear only at the base. And for another paper, which I will not describe here, so we looked here at the first part. Calcium goes. The first one arrive here, trigger calcium-induced calcium release, and then you have the release. But then the next question was, how does spine apparatus get refilled? Because if each time you have a synaptic input, it is depleted, then how it can be refilled? So this is another story which has to do with store-operated calcium entry, which again we found to be clustered near the membrane, and this is a way to refill calcium. So this is another story which I'm not going to describe. So extreme statistics is relevant, and the message from this paper is that the condition from extreme statistics is teaching us something about molecular organization. Okay, so if I have some time now, I can just describe a little bit of math to enter into the questions. Okay, many years ago, we worked on narrow escape, which is a time for a Brownian particle to reach here these narrow windows, dimension 2, dimension 3. And if you have been following this has to do with the surface here, because most likely the particle, the average time, will see on the domain, and log of 1 over epsilon. Epsilon is the size. And here in dimension 3, it's 1 over a. And if you do the expansion, you have the mean curvature. But that's mostly the leading term. V is the volume, the diffusion coefficient, a the size of the hole. If the hole is an ellipse, you see the ellipse, which is because of the property of Brownian motion. Okay, for the fastest particle to arrive in a domain, you look at the first one, you take the minimum of them, and what is very beautiful for the theory is that this is connected to n times the survival probability for one of them. And then you can do the theory. You have to solve. So if you're interested in calculating the mean time for the first one to reach a small hole, you start then with a diffusion equation, which give you, it's basically the Fokker-Planck, with the correct boundary condition, reflecting and absorbing at the narrow place. And then the distribution of the first time can be connected to the distribution, the survival distribution, and the flux to arrive. That's what is written here. And basically, you can connect this probability to the solution of this equation. And what you have to do is to solve this equation when n is large. And for this, you can use classical. I mean, it's not classical. You have to go into the mass of integration. But basically, in dimension 1, you solve it. The solution is explicit. It's the difference of two Gaussian. This is survival probability. You have here this Gamble law where n is the number of particles. And you get that the time is something of log of n. If it's an interval, it's the same, so it's not very interesting. If you have a drift, you see where the drift comes. It's log of n exponential a for a constant drift. So there is a connection between here, the drift, and n, if you are interested in this type of analytical result, which show the dependency with n. In dimension 2, again, the time it takes for the first one to escape has to do with the geodesic, the distance here from the source to the absorbing window, and you have log of 1 over epsilon. So you see the information about the geometry in log of log, which is very negligible, but still, this is interesting. This is dimension 2 and dimension 3. Again, you can see some of this information. And again, you have here the geodesic, the shortest distance. Epsilon is, where do we see epsilon here? Yeah, epsilon is the size of the hole. Okay, so now if I have some time, then I will stop. What happens if you don't have a delta Dirac at one point, but you have a distribution? It can happen because sometimes you have a first process that equilibrates quickly where you have a tail. And the question is, then if you have a tail, you should have a molecule that could be, even if it's rare, it could be still close to the target. And how does that affect? Well, if it's a delta Dirac, it's log, it's just what we said. But if it's suddenly you have a uniform distribution, it's 1 over n squared, not log of n. Then if suddenly you have something of x exponential minus bx squared, you have 1 over n. And finally, I think this is interesting, if you have something of this shape, x to power alpha with b, you can see where alpha and b enters, and you don't have any more log, but you have a power law. Okay, I think this is what I wanted to say. Yeah, what is large n? This is a fantastic question. I'm not sure I have time to discuss this, but maybe you can see that we want to connect the probability that something will happen with the fact that you need to have an estimate of n. And here what you can do is if you have a probability that one of many particles will reach 1, 2, 3, and so on, when p is small, this is the probability to access, and n is large, you have n, which is of order of 1 over p. So what is that? You have to calculate the probability to find something. Now, how do you calculate this? Well, if you have many windows, it can be one of them. And then this is the probability to hit one window when you have many windows out of those manys. If they are hidden in a cusp, it's more complicated because of the cusp. But this shows that if you have the same windows, basically the n that you need is of the order of windows. And if it's a killing, it's more complicated. Okay, I wanted to discuss how you accelerate simulations. I think this is, I think, I don't know if I'm done with time. Or shall I continue? Or okay, so I will not discuss this, but we can accelerate simulations. Yeah. No, that's it. That's it. No, okay. I wrap up. I'll finish here. n large could be a problem. Asymptotically, when n is very large, is a problem because, for example, if you think about senescence, if you have to go from this to there and you divide, but sometimes you can elongate, and if n is very large, you don't see the elongation. You arrive and it doesn't matter the dynamics. After some years of thinking, maybe n large is not okay. There's an intermediate regime that's the dynamic sees, for example, here, the time to senescence sees some of the dynamics, which you don't see in the limit n large. I can explain for those who are interested, although I'm leaving quickly. I thank you for your attention.